Welcome to the Theoretical Neuroscience podcast, now powered by the transmitter. I am the host, Gaute Einevold. The so-called central doctrine of molecular biology says that there are genetic information stored in the DNA molecules of the nuclei of our cells are transferred or transcribed, as it is called, into mRNA molecules containing the same information. These mRNA molecules provide the recipes that the ribosomes, tiny molecular machines, use to make the set of proteins needed for the cells to work. A protein is then made of a long necklace of so-called amino acids. When I learned about this back in the days, I got a mental picture that proteins, once produced, are stable, everlasting structures like the plastic molecule models we are shown in school. But this picture is incorrect. Like castles made out of sand on the beach, wear and tear destroy typical proteins in a matter of days. This represents a problem for neurons, as they need the right ion channel proteins to be present in sufficient amounts at the right place, for example at distant synapses, for the neuron to function properly. Proteins must thus be produced continuously, and this requires energy. The cell has two options. The first option is to make the proteins in the soma just outside the nucleus where the DNA is stored. The problem with this is that it takes energy to transport the proteins, and if a lot of proteins disintegrate on the way, it is wasteful. The second option is to transport the mRNA to the place where the proteins are needed, maybe at the synapse, and produce the protein there. But this also costs energy, especially if the mRNA molecules are unstable, so that a lot of messengers disintegrate on the way. The brain seems to be optimized for efficient energy use, using only 20 watts or so, and the natural assumption is that the molecular machinery also is energy efficient. So what strategy is best for a particular protein in a particular neuron? Centralized or decentralized protein production? The answer depends on the situation, how large the neuron is, how long-lasting the proteins and associated mRNAs are, the time and energy consumption related to transport inside the neuron, and so on. When many factors contribute and numbers matter, mathematical modeling is often called for. Today's guest, Tatjana Tchumatchenko, and her group made a model to study this problem. Tatjana is a computational neuroscientist and professor at the University of Bonn in Germany. In a paper published last year, they showed that their model, when asked to optimize for energy efficiency, indeed predicted experimental findings regarding the spatial distribution of proteins and mRNA molecules. In the podcast, we discussed the problem and how Tatjana's group designed and used the model, and the new predictions and questions that came out of it. Towards the end, we talk about something very different, namely about how it is to start a computational neuroscience lab, and in particular the challenges of reorienting a research profile. Before we begin, it's a good time to remind you about the podcast homepage at theoreticalneuroscience.no. There you can find information about all the episodes, as well as links to all the podcasts, and each episode has its own information page with links to papers, books, or websites that are relevant for the podcast. I'm happy to tell you that the podcast is now powered by the transmitter. The transmitter, which can be found at thetransmitter.org, is an online publication dedicated to neuroscience news, analysis, and commentary, aimed at researchers and people across the neuroscience community. You should check it out. The transmitters support the podcast both financially and by showcasing all episodes on their website as they are released. I'm very happy about this new collaboration, as my hope always has been that eventually everyone in the neuroscience community should know about this podcast, whether they would like to listen to it or not. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode via the Patreon website. And note that there is a 7-day free trial period when you sign up. As mentioned, I also have a YouTube channel called Theoretical Neuroscience Podcast, where you also can watch the videos. I really appreciate all of you who are already Patreon supporters. Thanks a lot. I will be most grateful if more join in to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Tatjana. Yeah, so welcome to the Theoretical Neuroscience Podcast, Tatjana Tchumatchenko.
Hi, welcome. I'm excited to be here.
Yeah. So, very good. So well, the background for this podcast is this, I would say, this really cool paper that I read from last year from your group, How Energy Determines Spatial Localization and Copy Number of Molecules in Neurons. Because I think although most computational neuroscientists have focused on, I would say, signaling and coding and electric stuff, I think to the extent that we know anything about energy consumption, we have thought that sort of like the main energy consumers in the brain, maybe, or in the brain, is sort of like ion pumps at the membrane that sets up these concentration differences, and also the expense of making these synaptic vesicles to use in the
Synaptic cleft.
in the exactly, so synaptic vesicles to make the communication. But then you sort of argue, I guess, the point of the paper is that there is also a big energetic cost in sort of just making and transporting these proteins around. For example, the ones that make a neuron with all the ion channels and stuff.
Exactly. We now know
Mm.
that there are tens of thousands of different proteins,
Mm.
but what people appreciate less is that all of these proteins have a half-life. They
Mm.
don't live as long as the whole organisms live.
Mm.
And for the proteins in the mouse, but also in humans and other species, we know that a typical lifetime is on the order of days. Give
Mm.
it maybe half a month, but that's about it.
Yeah.
Of course, there's this exponential decay. Some might live longer, some shorter. That's an average, right? But the expectation that a single protein, when it is made, will survive even half the lifetime
That's
of an
true.
animal, that's an exaggeration.
I think that's
Also, mhm,
yeah. This is sort of true. I think this is also how you typically think about this thing. You make a protein, and then you'll solve that forever.
exactly. We
That
think the
it's
cell is made and that's it. Then
Yeah,
it has to survive.
that has been the sort of like at least in my head, like the simple picture.
Yeah.
So,
And we
so
know
how?
that
Yeah.
there's like a big turnover process that needs to be
Mm.
managed, because if you
Yeah.
imagine 10,000s of different protein species need to be
Mm.
produced, they need to be trafficked, and then they need to be degraded, and then the cycle
Mm.
starts over again. And
Yeah.
that's asynchronous, and everything needs to be titrated just so to make the neuron functional.
Mm. So, okay. So, what first sort of pushed you to focus on this problem? Do you have any? I mean, where are you now? You're
In
in
Bonn,
Bonn, right? Yeah.
exactly.
What it's called? Mm.
I'm a professor of computational neuroscience of behavior, actually,
Mm.
at the medical clinic in Bonn. That's part of
Mm.
the University of Bonn. And throughout my career, I have been focused on how circuits work, how neural circuits synthesize computations, how neurons communicate through synapses in a way that enables this computation to emerge. And then I noticed that there is a big chunk of neuronal life that we don't know much about in terms of computational terms.
Mm.
We think a synapse is made and then there is this plasticity pushing it up and down. But it turns out a synapse is made of proteins, and mRNAs need to be produced to produce the proteins. And then I
Mm.
wondered, how does that work mathematically?
Mm. So maybe so that's just like take a little course in this. So you have the DNA is in the even a big neuron like a pyramidal neuron. All the DNA is only in the soma,
That's correct,
right?
in the
So
nucleus.
in the nucleus, exactly in the soma.
Hmm.
And then this thing has to be transferred or made into this messenger RNA,
Yeah,
mRNA.
exactly.
Yeah, and
That's
that's
exported out.
yeah.
That's
Or it
exported
could,
out
yeah.
through
It
the
doesn't have to
nuclear
be.
pores. Hmm.
I mean, I guess it could sort of the mRNA could then be made into these proteins in the soma.
That's correct. That's
Yeah,
an
so
option.
that's one option. And then to make these proteins, you need something called ribosomes, right?
Hmm. Exactly. The ribosomes need to be assembled. They are consisting of multiple units, and when these units come together, they lock onto the mRNA, and then
Mm,
they are able to transcribe one by one the nucleotides and attach the resulting amino acids to each other to produce that protein chain that we know about, and then it needs to be folded and released into the cytoplasm.
yeah. So then the key thing is that this is mRNA, this for production of the protein based on mRNA could in principle happen elsewhere,
Yes,
like in the synapse, right? So then it's like a trade-off. Should you send the messenger, the mRNA, or should you send the
exactly.
protein? So what should be transported? But and then you also have something called mitochondria. What do mitochondria do?
The mitochondria are the energy factories of a cell. They produce, in the simplest scenario, they produce ATP to
Mhm,
to enable all the other cellular functions that neurons
mhm,
need in order to function. For example, in the synthesis of protein, there are several steps that take up ATP and GDP, for that matter, and GDP is another way to provide energy to the cell. But we have focused mostly on the ATP, on the ATP need of a cell in our modeling. So an ATP is a molecule that is required to facilitate processes that otherwise would not happen. So it's like the way we use energy in our home, right? The fan that we need when the heat is high, it won't turn up on its own. You need somebody to push the motor to turn,
mm,
and that energy needs to be produced by, in our case, solar panels or other factories. But in the cell, somebody needs to help those processes along, and there are these ATP molecules that are required for that. So
And
there are
that's
some processes that happen on their own, and
mhm.
other processes need energy to be started, and then to be kept on for the cell.
So that's like so then you have like this ATP as sort of like the some sense like the molecule as an energy currency, and the ATP. This is the reason we have to eat, right? We need food to produce ATP,
Yes,
right?
exactly.
Yeah. So
There are
that's like
several
the,
processes that synthesize ATP,
mm,
but at the heart of it, it's glucose. When there is a
sugar.
glucose cycle, and when we have one state of glucose, um, so yeah, maybe we cut this part out because I'm
No,
not a
no. No, that's fine. Let's just
biochemistry.
go ahead. We can have sugar. That's fine because it's good to have this the big picture clear for the readers because this is something that typical computation neuroscientists are sort of like in. They don't necessarily. Well, they do. You don't typically learn about it in the computation neuroscience course, but I guess sort of you get these sugars like in glucose, and this
Mhm.
is then after some biochemical signaling pathways and processes and big like metabolic network
Exactly.
turn
In
into this
the end, the currency is produced, the ATP.
ATP.
Um, yes.
Yeah. So very good. So then, uh, and then there is this. I guess if you sort of then, so what's the experiment in order to test sort of like, well, I guess okay. So back to the mitochondria. The mitochondria is sort of these energy conversion factories
Energy synthesis machine for the
synthesis,
cell.
exactly, making these ATP molecules, right? So, and these mitochondria can be all over the neurons also, right? They are all over the neurons. So it tells you something, presumably that in a region where you have a lot of mitochondria, you might need a lot of energy.
That's a hypothesis, right? It
Hypothesis
turns
at least.
out when we looked into the data to help us understand how mitochondria is distributed in the cell, it was
Mm,
not as abundant as we thought it would be. And then we
mm,
located some papers to tell us that soma is a region where mitochondria are abundant. However, in the
Mhm.
dendrite, there is another abundant concentration of mitochondria, and it
Mhm.
decays as a function of distance from the soma.
Mhm.
However, that's actually part of the active research in my lab to figure out how exactly that distribution of mitochondria is arising and whether
Mhm.
it's dynamic. So there are many things that we don't yet know.
Mhm. Ah, so you do experiments in your lab also?
No, no, we are a purely computational lab. We
Okay.
analyze other people's data
Yeah.
and we collaborate with colleagues that can measure them.
Okay. Are there also colleagues at in Bonn that you can collaborate with? Or?
Yes, there are wonderful collaborations also in Bonn, more on the protein level, on the synaptic level, but there are
Mhm.
also groups measuring the biophysics and the biochemistry, the processes that take place in order to synthesize ATP in the mitochondria, the anatomical shape, how these signaling cascades are turned off and on to synthesize
Mhm.
more or less ATP. Those things are investigated in Bonn. That's right.
Mhm. So okay, so then I guess if you then look at these data, I guess and this is something they call it what's it called? They have this fancy name when you measure the where the proteins are in the cell. They don't call it measuring protein; they call it proteomics or something.
Yes. So
Yeah,
proteomics, and I'm a computational person, the way
yeah.
I understand this is there are multiple parameters to be measured about the
Mm-hmm.
proteins in a cell. One is you want to know how many are where. So you want that density as a function of distance and time.
Mm-hmm.
And you want to know what processes synthesize proteins. So where exactly is a single protein is produced, how long it lives, and how many there are per species, and how many species in total. So you can take up a whole cell and take and calculate a cell proteome with all compartments included, or you
Mm-hmm.
can take a specific compartment, let's say the soma or the nucleus, and not the nucleus. There are no proteins that are helping the synapses function in the nucleus. But you can take different compartments and look compartment by compartment, how many proteins they are, what species you find where, how many copies of each species, and then you can look into the dynamics, how this um,
Mm-hmm.
whole proteome is changing as a function of time.
Yeah, because a proteome is like the collection of all proteins, right? That's like
Exactly,
the
it's like the fingerprint. The
of all
um,
proteins. And then you
exactly.
have also this something called even fancier name transcriptomics. That's where this
Where the
mRNA.
mRNA is,
Yeah,
exactly.
exactly.
Every
So
protein is made of is produced from the mRNA. It's
mm,
transcribed from the mRNA, so you have a one-to-one. Um, uh, there's there's a precursor for each protein,
mm,
and the question is, how many precursors do you have? How long do these precursors live? And it turns out that the precursors live way shorter than the protein themselves. A
ah,
typical mRNA has a half-life of a few hours.
so
So
the
you
messenger does.
produce it and then you turn around and it's gone.
Oh,
And
really?
you need to produce again.
Exactly. So the messenger boys doesn't last very long. So
Yeah.
you better produce it before they vanish. So okay, so then and then if you then look at, of course, now you have all these techniques for measuring these the transcriptomics and the proteomics, or the transcriptome and the proteome, I guess. And then it turns out that if you look at proteins, some of them have a lot of this mRNA spread out all across, also, and then the neuron also at the
Hmm.
distant places,
Yes.
implying that you actually these factories are these protein factories are up there
Hmm.
also.
In the
And
dendrite, yes.
some are not, right? Some have like all the mRNA in the soma region, indicating that that's where the proteins are made for this particular no, protein, right?
Hmm.
So this, I guess, you as well, you being a physicist, then of course you're looking for a
Per rule.
principle,
Why?
exactly, some kind of global rule, exactly, principle
Yeah.
for why is it like that?
Yeah. And when you look just at the statistics, when we started, we found out that many protein species localize the mRNA to the dendrite, however, not all. So
Hmm.
if you take a class of synaptic proteins, PSD, for
Hmm.
example, has a lot of local mRNA, But other proteins that are equally important
Yeah.
for synaptic function, they have the somatic mRNAs. The
Yeah.
question is why? What decides
Mm,
which way it's going to go? And is it
mm,
always just
uh,
one decision and then the mRNA stays there?
yeah.
How is
Mm,
this decision process
mm,
mathematically made?
yeah. And then, of course, then I guess you also had this assumption that this cost energy. So actually having as a, well, of course, being a physicist, a computational neuroscientist, you want to make a model to try to
Yes.
understand this, and then also you had that this idea that this is so expensive energetically that
Hmm.
they probably that the neuron is probably has developed in order to sort of minimize the energy consumption
Yeah.
associated with having all the proteins or the ion channels at the right places and so on.
Exactly. When you write a differential equation that governs the density as a function of space and time, you realize that there are parameters in it, right?
Mm,
And you can solve this equation and you just can scale up everything. You just multiply everything by a factor of 10 and it will be
mm,
just as good because
mm,
the density is not constrained by the amount. It's
mm,
constrained by the distribution, right?
mm,
And then the question is, who tells you how many to produce? Why don't we just multiply everything by 10, for example?
mm,
What's the upper ceiling? The lower
mm,
ceiling is relatively easy to determine. You say, if I'm supplying the dendritic tree and the synapses of the dendritic tree with proteins, I would just say every synapse needs a minimal number. That's a
mm.
reasonable assumption. But
Yeah.
then computationally, you need also the upper ceiling because
Mm,
again, you can produce more than you need and there's nothing stopping you. And that's why I
mm.
thought maybe there are energetic constraints to limit the total. And that's
Yeah.
plausible, right? You also
Yeah.
don't buy
No,
everything
no.
you want. There is
No.
a money
Yeah,
ceiling
that's
for
true.
what you can afford. And that may be also going on in the cell that I thought.
Yeah, exactly. No, that makes sense. That you certainly don't want to waste food, right?
Yeah,
Especially in times of where food has been a limiting factor,
yeah.
and I guess that's well, long part of human history often, and
Exactly. And we know
yeah.
that many processes that make a protein and deliver it to the particular synapse of interest, they take up energy.
Mm,
The synthesis of mRNA per nucleotide attached,
mm,
that's an energy cost that's required. Per mRNA trafficked, you incur a particular trafficking cost. Per synthesis of amino acid attached to
mm,
a protein chain, there is an energy for that. So there are several processes, and there are way more.
mm,
The folding of a protein also takes energy in some cases. So there are certain processes that are for sure required to make sure a protein can show up at the synapse. And the question is, how do you decide how many to produce and how to weigh these costs against each other? Do I
mm,
produce more proteins and then hope that some of them make it to the synapse? Or do I help them along to make sure more of them arrive at the synapse? That's a question of paying for the transport. Do you afford it or not? Do I
mm.
need a taxi to get to the synapse? Yes or no?
Should I just wait for exactly? Should I walk or should I take a taxi?
Yeah.
Yeah. So, and but you talk a lot about the synapse. Of course, there you have like these well, required ion channels, but there's also, I mean, in the axon you need these sodium potassium channels all over the place, right? So
Yeah.
it's a whole neuron needs these ion channels, not only the
Yeah,
synapse, right?
that's a super interesting question. We know that the trafficking,
Mm,
the motion of the motors, the kyanese and dynein that walk along the microtubule
yeah.
in the axon is very different, very much directional than in the
Uh,
dendrites. In the
mm,
dendrites, it's more of a tug of war situation where you walk
mm,
2 steps forward and 3 steps backward and so on. But
mm,
in the axon, it's much more forward motion based on the experimental data. And we expect some differences between these compartments, but there are many, many questions that we don't yet understand. However,
mm,
we currently know that some mRNAs are synthesizing proteins in the axonal tips. However,
mm,
how many species do that? All of these statistics is still to be discovered, amazingly.
yeah. Mm. So, okay, so now to the model. Of course, you a good thing with like dendrites, at least when you have a focus on the like intracellular focus is that they are essentially linear. I mean, except if we like you did,
Hmm.
like what I also would have done if I start with a linear segment. So a linear, so you consider like
A
a
line.
linear dendrite
Hmm.
of a certain length,
Exactly.
and then you also, the important thing is that you have, in addition to the dendrite, you also have these spines, which are attached to the dendritic shaft. So what is a spine?
A spine is basically a protrusion. Excitatory spine is a protrusion. Inhibitory synapses on the postsynaptic side don't have the spine, so it's a little bit harder to define. But excitatory spines are basically like a little pocket of membrane that is protruding from the dendrite itself.
Uh-huh.
The
Ah,
spine neck, the piece that is connected to the shaft, can be of different lengths. And then there
mm.
is this like a pocket that can harbor a PSD, for example, or other signaling
So
molecules.
PST particular.
PSD, postsynaptic density
Okay.
proteins
Okay.
that
Mhm.
anchor the receptors in place.
So exactly. So so you need to have a certain sort of like density of the right proteins
Hmm.
inside the spine in order for this spine to function as it should, like to have this synapse.
Exactly.
So the three main dynamical variables then which you have in in your model is the concentration of a protein on
Hmm.
the shaft,
Yeah,
and the concentration of protein at inside the spine, and then also the concentration of this mRNA producing this particular
exactly.
protein, right?
Maybe we start with the differential equation for the mRNA because it's very simple.
Yeah,
Many
let's do that.
mathematics and physics students
Yeah.
do that all the time. They start with exactly this equation when they model anything. There is a diffusional term, the
Mm,
second derivation with respect to space,
mm,
acting on the density of mRNAs. And in front
mm,
of it, we have a diffusion constant that
mm,
basically tells you um, how how diffusive that mRNA is,
yeah,
how much distance can it cover on its own.
mm,
And then there's a degradation terms. There's a minus degradation rate times what you already have. And
mm,
a source term at the soma. That's
yeah,
where the mRNAs are produced. They have a particular rate of production, a particular rate of degradation, and a
mm,
particular motion characterized by the diffusion constant. And that's an approximation. There
mm,
might be a little bias, but we neglected it because we didn't see it really in the data. So mRNA is a simple, really vanilla differential equation
mm,
with a diffusion term. And the proteins are a little bit more complex because they have this uptake term into the synapse. And the way we designed the uptake term is we said there will be a flow of proteins from the spine shaft into the synapse, into the
mm,
postsynaptic density, if the number of slots reserved for that protein species are not
mm,
yet filled. So as long as
yeah,
there is a mismatch of how you want and how much you have, we will
mm,
we will give you some. And then there will be a passive diffusion out. So it's
mm.
something that basically happens
Out
randomly
out of the
out of the
spine.
synapse, out of the spine. Exactly, there is
Mm,
a passive diffusion out, and a term that basically depends on how filled you are already.
mm,
Those are the
yeah.
two terms that govern the exchange, and the rest is very much standard. Again, the diffusion along the shaft, degradation and synthesis. Synthesis will depend on where the mRNAs are, so
Mm,
exactly proportional to the mRNA density, and
mhm.
that's it. There is not more magic in it.
No. And then this ends up, well, you have some, maybe you mentioned it, but you also, this active transport. I remember this is something which I remember the first time I saw like animation of this active transport. You have some kind of particular transport proteins
Hmm.
that are carrying things essentially on their back and
Yeah,
walking
the
up this like
kinesin
internal,
motors, right?
yeah,
Yeah,
walking up inside on this almost like a railroad track inside the cell.
yeah,
So this is sort of like active transport, which is needed
exactly.
to get things to the, because diffusion, the free diffusion is quite slow, and not because it's not directed anywhere. While active transport is faster. So it's
supposedly
maybe
faster, right?
yeah.
However, what we already know when we look into the data of how the cargo is moved along the microtubular tracks in the dendrites is that the plus and minus ends of these tracks are
Mm.
basically equally distributed on each end. So,
Ah.
so yes, there is a directed motion, but
Mm.
when you track individual particles in the dendrite, you find out that they don't always walk in the same direction. There's a lot of back and forth. And
So,
that's why it's directed, yes, but only for a little
so
while.
I see. So you have these busy workers, and some of them are half of them almost are just going in the wrong direction. So it seems like a very,
Very
very limited.
inefficient
Yeah.
process. And then you can ask the question, which proteins are actually transported actively? And that's where things get interesting.
Yeah. Mm. So
They,
how did you model? So how did you model this active transport
yeah, with a
in? Yeah.
diffusion term. In the
Yeah.
dendrites, simply with a
Okay.
diffusion term.
Okay, but then it's a faster diffusion then.
Exactly. The amount of that diffusional constant will depend on
Uh,
how strong, where you diffuse. Do you diffuse on the
yeah.
membrane? Do you diffuse in the cytoplasm? Do you diffuse along the microtubular tracks through that back and forth motion? That value will be situation dependent. But the fact
Mhm.
that it is the diffusional constant, we saw that's the first assumption that we can live with.
Mm. So okay, so then you have this. It's like one spatial dimension that's like up or down the dendrite essentially, and then you have time. So then you end up with like what's it called, a partial differential equation. It's the differential equation both in like space
Yeah.
and time, which is sort of incidentally looks a bit like the cable equation, right? It's
It's
like
very much so. Yeah.
and almost the Schrödinger equation for
Yeah.
quantum mechanics also is like this, is like if equation and that shows up many places in
Yes, if you
in
master
inside.
this equation, you're pretty good in
Yeah,
computational
so
neuroscience.
that's pretty good. Okay, so that's cool. And then, of
And
course,
then you
then
close the dendrite. That's where things get interesting too. If you
when you
leave the
what?
dendrite open, there are
Yeah,
boundary conditions, right?
yeah,
There are
that's
boundary
true.
conditions. If you want to conserve the amount of proteins you insert, you
Yeah,
need to make sure they don't leave at the end of the dendrite.
that that would be very wasteful. Yeah. So there is a reason we have a closed membrane. Yeah.
Exactly.
So that's well, that's another reason we have a closed membrane. So that's good. So then, and then of course you need to, no. So then you look for, in the first case you look for what's called steady state solutions of this equation. What is a
Exactly.
steady state
We,
solution,
we
and why do you
basically
focus on
say
that?
that there is no dynamics. There's a
Mm,
first approximation that the
mm,
differential with respect to time is zero,
mm.
and everything on the left hand side of a differential equation, and mathematicians put the temporal evolution on the left hand side, and you just kill it. It's
Yeah,
gone.
it's
and
zero.
and then you solve what
Uh,
in a data situation would be considered an endogenous average distribution.
Yeah. So the point is that you look at this. The idea is that, well, for this neuron to work, you have to have the right proteins in the spines, a 95% filling at
Yeah.
all times.
Yeah.
So you just look at, so then you have to, and since they degrade, you just have to have this
Yeah, you need to sort
constant
of
flow.
balance the synthesis with the
Yeah.
degradation, ideally in all places to make sure every synapse is supplied.
Mm,
And then
mm,
you fill up the dendrite. You set all of the parameters the way that you would think are biologically plausible, and
mm,
then you upregulate the copy number.
mm. What is a copy number?
A copy
That's
number is how many copies, so how high do you want that density to be? Because the
okay.
density is proportional to
So
the
I see.
total number
I
of
see.
proteins, and density times the total is how
That's true.
many are in any particular place,
Mm.
and that's where you can regulate
Copy
the
number.
amounts.
Yeah, exactly. So if you ordered three pizzas, the copy number is three.
Three exactly.
So this is
But
this is
if
also what it is. Yeah,
exactly, but if you say there is on average, like the density of pizzas is
yeah,
zero point 0.5, that's
yeah,
great. Between your
yeah,
neighbors and you have zero
yeah,
point 0.5 and the neighbor
yeah,
has zero point 0.5. But how many pizzas do you actually arrive? It depends on how many are produced, right?
yeah. That's true. Yeah. So okay, and then of course this a key thing here is sort of, so this is a model that you think should sort of like well explain why some proteins are produced mainly in the soma while others are produced out in the distal areas at the synapse.
Hmm.
And so what is different here is the essentially the parameters, the parameters of these different proteins. So what are the key? So you have like
Yeah,
the
there are
half
several
lives.
parameters.
Yeah,
Exactly. There are
yeah, what,
several parameters
yeah,
characterizing this pair. It's always
mm,
a pair of mRNA and protein.
mm,
And then you have, as a theoretician, you now have to set the parameters. And the question is, where do you get them from?
yeah,
And the natural answer is you need to look at the data. So you need
yeah,
to scan the literature. You need to talk to experimental colleagues to determine
mm,
each one. And then
mm,
the question is, okay, you have your individual set of parameters figured out. Then you can model only one protein, right?
mm,
One set of
mm,
parameters, one protein. And we thought,
mm,
well, we can't possibly go this way for tens of thousands of proteins. We need some parameter distribution where we can
mm,
sample parameters, right? We just
mm,
draw randomly for now
mm,
our parameters and
mm,
see what we get. You can also draw parameters in a correlated fashion, or you can
mm,
draw them independently of each other. That's a question to be determined. We said
mm,
as a first start, we're going to draw the parameters independently from the parameter distribution that we take from the data.
mm.
Half-lives are measured wonderfully. On mRNA half-lives and protein half-lives, there are many, many papers you can
So,
draw the
so what are the
distribution from.
typical time scales of like a half life of a typical? Well, what's a typical protein? I'm not quite sure if that is even makes sense to say, but like minutes, hours, or
For protein, it's days.
days. Okay,
Days.
that's good.
I think 5 to 10 days on average, depending on which data set you take, is a good
Yeah,
estimate. For
yeah,
for mRNAs, depending on the data set you look, a
mm,
few hours, maybe
few hours.
5 to 10 hours is an average.
Yeah. Okay. So and then of course your model applies to any kind of I mean, you could apply it for any kind of assume all kinds of parameters, but then later you sort of start. Okay, what should I given these parameters? How should I minimize
Hmm.
the energy? How should I put things?
Yeah,
Yeah, how should I sort of like? Yeah, where should I do things in
yeah.
order to save energy?
Exactly.
And
And
then you,
you sampled your parameter set. You
yeah,
had a random draw. Each parameter is set to any particular value that's consistent. And
mm,
then the way we address this to determine which mRNA distribution you will get is you
mm,
calculate the things twice. You
mm,
calculate things under the assumption that all mRNAs are in the soma or in the
mm,
nucleus that's contained in the soma,
yeah,
or you calculate it under the assumption that the mRNAs are distributed in the dendrite. And
mm,
then you compare the two numbers and take
yeah,
the cheaper one.
yeah, of course. So this is sort of like some company deciding where to put factories, I guess, and produce things. Because I
Yeah.
guess if you are sort of like, for example, if you're sort of running like a takeout pizza chain in Germany,
Hmm.
it wouldn't make sense to have all the pizzas put in the oven in Bonn and drive across the country with it because
That's
it
the
would be,
logic. Exactly.
yeah, it would take too long time to get the pizzas to Berlin. So that so it will be old, right? The half life of pizzas are maybe like hours. I
I
don't
can
know
tell you it will depend on the half life of the pizza. If you're willing to eat a
exactly.
cold pizza, it's perfectly fine.
Exactly. But while a car, right? The half life of a car is like, I don't know, years,
Yeah.
right? So then you can have a very centralized factory. I guess that's Wolfsburg or something, or some of these where these
Yeah.
German factories are.
Also, exactly, half life is one important parameter that
Ah,
the theory also surfaced, unsurprisingly. The other parameter is how much the trafficking costs
yeah,
and how much the synthesis of a protein costs. If it's something
yeah,
super precious, you better
yeah,
worry how you shuffle it
yeah,
around. But if
mm,
it's cheap in the sense that it's maybe a short amino
yeah,
acid to produce, it's it doesn't cost so much if you make a
no,
mistake.
yeah. I guess because I guess that's sort of if you look at the like a lot of the production of goods in the West that consumers in the West buy comes from China
Yeah.
because the production price pays is more expensive or
S-
covers more of the cost than the production.
Yeah.
which are on these big ships, which I think is quite cheap actually.
Yeah. So if the
To fly.
transport cost is cheap, then
Yeah,
the
then you do it
distribution
forever. Oh,
of sources is different.
yeah. So then, but you need, in order to keep like have this energy bookkeeping in the cell, you need sort of like some estimates for how much ATP, how much of this joint energy unit is sort of involved in the different processes. So I guess uh,
Yeah. And we have a
transcript.
breakdown. We have your
Yeah, exactly.
itemized list, how things
Exactly.
cost. And
So that's
yeah,
sort of like this is what economists do, I guess, in companies.
yeah.
Yeah,
And we
they
found
set
out
up.
that translation is actually on the top of the list. So
Yeah,
if you can
that's
save
okay.
just a little bit, if you produce just a little bit less,
Yeah,
it makes a big difference.
yeah. Translation. So what that includes? Okay, so you have first transcription. That's making
mRNAs.
nucleotide mRNAs. So that's like I said, 2.17 ATP per nucleotide per like
Yeah,
yeah, per nucleotide, like in the uh, basic building block of the mRNA. Uh, Almost like DNA, but not one of the bases or changes or whatever it is. So, so that means that if you, it of course, longer transcripts
will cost
uh,
more.
cost more to make, right?
Yeah,
And what I also learned, which I didn't know, that they have like they don't. Of course, you code for the you need to code for the proteins that are going to the amino acids, like the beads in this chain of amino acid that makes the protein. But they also carry with them a bunch of like non-coding regions. So they
yeah,
produce not only the recipe,
yeah,
they produce, well, they you have to produce more and carry this whole thing
exactly.
up with it.
Yeah, the mRNA, we think of it as a theorist. I think of it as a line, but it's actually not a line. It's folded in very interesting ways, and those parts that are non-coding, they
Mhm,
determine how many other mRNA binding proteins can come in, because you need them to start the translation. You need them to make sure that mRNA is not transcribed, not translated at some point in time. So it's about the regulation where things should be translated and not. And for
mhm.
that, you need machineries
So,
to dock
yeah,
onto the mRNA.
because translation, that's making the proteins
That's
from the
making the
mRNA.
proteins,
Yeah.
exactly.
And you say this costs about, so that's like more than twice as expensive, or at least if you compare per amino acid,
Yeah,
that like it's that's 5 ATP per
yeah,
amino acid according to your paper.
yeah.
So this is sort of yeah. So I guess if you, yeah, so that's that's typically what dominates the cost when mRNAs are in this, when everything is made in the soma.
Hmm.
Is that what you say? Yeah.
Yeah,
Because then you don't yeah, I guess because with mRNA not lasting so long, if you do it in the soma, then you sort of you don't need to produce so much, I
yeah.
guess.
Like
Or
viewed
you
at
don't
it mathematically is, if you have a short-lived mRNA and a long-lived mRNA, you have two examples,
mm,
one short, one long-lived, and both are in the soma.
mm,
You produce less protein for the short-lived, right?
yeah,
And if that protein happens to be really expensive in
mm,
synthesis, then you won't have enough in the synapse. So that
mm,
short-lived um, mRNA,
mm,
if it is if it is synthesizing the proteins in the soma, it should
mm,
upregulate its translation a little bit so that that decay en route is compensated for.
mm. Yeah,
And
exactly.
if you are producing in the dendrite, there is a shorter distance to the synapse of reference, and
Mm,
therefore you might produce a little bit less because there is
mm,
less decay on road. And that
yeah,
difference in
exactly.
production is making things more or less expensive, because
Mm,
translation is the largest budget item.
mm. Yeah, and okay, so then and then you also need to transport these things if you don't want to, at least I guess. Yeah. So if you make everything, if the factory is in the soma, then a lot of things has to be transported, and that
And that's a careful
and
budget. Per
yeah.
protein, you need to look at how many, what's the half life of mRNAs and proteins
Mhm.
of my
Yeah.
species? How much would it cost to to move my mRNAs. What's the
mm,
amount? And then
mm,
you carefully calibrate it for each individual parameter combination. And for some, you
mm,
determine, okay, maybe I leave them at the soma, and for
mm,
others, I would move them into the dendrite. But it turns out it's not always a binary decision. You can do 50-50, or you can do 90-10.
mm,
And that's where the interesting questions arise.
mm,
And also, if you determine that ratio once, you might revise your calculation based on plasticity or based on other factors. Maybe
mm.
ATP amounts are available less. You might change your parameter combination and revise your decision where to move the mRNA. So things are really dynamic in neurons.
Yeah, it's so you say it's dynamic. It's like it varies between proteins. Yeah,
It varies
it
across the protein species, and
yeah,
also potentially across the lifetime of a neuron or
yeah,
cell types, right? Some are
yeah,
shorter, some are longer, some have
exactly.
more branches, some
Mm,
are some demand more ATP, some demand less.
mm,
And that's where they have less available. So that's where you need to titrate the balance of synthesis and degradation in different ways per protein and mRNA.
yeah, and one thing that is, I guess, is not included in in your optimization is the making and the cost for energetic cost for making and sort of placing ribosomes.
That's
And
right. We
yeah,
excluded this because we
yeah,
thought that if the ribosome is doing its job, it should cost just as much, regardless where it is.
yeah, mm,
Now, that's an assumption because technically the ribosomes also need to be trafficked. So there is a
yeah,
little bit of a infrastructure
mm,
cost associated with
yeah,
dendritic synthesis.
mm,
However, if you save enough, it's okay.
mm,
but
yeah,
but the future models could go into this particular direction.
yeah. I guess also the making and the placing of mitochondria is also one thing that sort of
Yeah, that turnover
sort of
of
like
mitochondria is not so well understood
no.
because it also
Hmm.
takes ATP. There is
So
trafficking involved, and it's an organelle. It's a big structure. Proteins are smaller than
yeah,
the
because
the
it's actually
organelles.
something which is have their own membrane and stuff. It's their
Exactly.
own little yeah. So exactly. So there is sort of like this the cost of making the factory and getting power to the factory out where when it's distant is not directly included in
Mhm.
this particular at
In
least
this particular,
and
and
this
this is
model.
the
But
first model that quantifies
yeah,
this. If
yeah.
students and postdocs looking at this
Mhm.
podcast and watching this podcast, if they have ideas, they are more
Yeah,
than welcome to expand.
that's true. No, I think at least I really like this. At least from when you're training in if you end up with a one-dimensional partial differential equation, then it's you're not overwhelmed by complexity. It's actually possible to think about it, and sort of get some intuition about it. So I think it's you found like a very nice balance. I think between sort of like complexity
And
and
it
sort of
still
like
does the job, right? It can
yeah,
predict these ratios of somatic to dendritic mRNA quite nicely. So complexity should also come at their with some benefits, right? And
yeah. So, and then you had like, of course, now you want to compare it with experiments, and there's one protein called
kinase
CAMK2
two,
alpha.
yes,
CAMK2 alpha. Yeah.
it's a
So
plasticity relevant proteins. Many
yeah,
plasticity
so
models have it included. It's
you
phosphorylated,
need it.
dephosphorylated to facilitate plasticity. Yeah.
So you need it in, up in the synapse to have synaptic plasticity,
Yep.
and that's something we are
We
we
care
like.
about, right?
We care about here in the podcast. We are support synaptic plasticity here in the theoretical neuroscience podcast. So what kind of what kind of protein is this?
Um, it's one that has a half-life of about 8 days,
Mm,
and we know that the average dendritic spine, contains about 32,000 kinase 2 proteins.
okay,
That's
is
a
that a
high
lot? Is that
number.
a lot? That's a high number. Yeah.
That's a high
Mm,
number, and
yeah.
the particular kinase 2 protein contains 478 amino acids, which
Wow.
is a
Yeah.
longer chain.
Yeah. Okay.
And
Yeah.
when we ran the numbers for
Mm,
somatic synthesis versus dendritic synthesis, we came
mm,
up with the idea that the dendritic synthesis makes more sense for that particular protein. And then we
Mm.
went in and checked, and it turns out it's correct.
Yeah.
And for proteins, you need to make always a calculation which amounts of energy would you get if you would you have to pay. Not, you don't get anything, but you would have to pay if you synthesize them in the soma versus dendrite. And for that particular protein, it was 55% more efficient if the
Mm.
mRNAs were in the dendrite, which also
Okay.
tells you that if you are efficient, you should invest in that strategy
Mm.
that's more efficient. But if you make a
Yeah.
mistake and keep some mRNAs in the soma, it's still
Mm.
okay. But on average, it's better to move them.
Mm. Mm.
That's
Yeah.
that's why mathematically you should see mRNAs being trafficked to the dendrites in that
Mm.
particular species.
Mm. And that you also, as you said, you also see,
We also see exactly. That's
right? An
actually one
experiment,
of the
an
best
experiment, right?
characterized proteins in the dendrites of neurons. And there's a lot of data, including the lab of Aaron Schumer has produced several data sets that we found wonderful to analyze, where the mRNA distribution can be characterized with a particular exponential distribution, an
Mm.
exponential decay factor. We characterize also the protein distribution, and the two actually in our model could be
Mm.
predicted just by the fact that the energy consumption needs to be balanced. And from
Mm.
there, we could derive the particular shape of the spatial distribution for RNAs and proteins. So from the
Mm.
parameters of the half-life, of the synaptic uptake, copy numbers, and everything that we know that
Mm.
characterizes that species, we could derive how it's distributed in the dendrites. So we didn't fit it per se in that paper. We fit it in other
Mm.
papers, but here we didn't fit anything. We just predicted based on parameters.
Yeah. Cool. And then you, of course, when you have a model, and then you can do many more things with a model that you can easily do in experiments. At least it's like less work. You can get the computer to do the work for you. So then you wanted to sort of look for some like general rules, and so you generated like more than 2,000 sort of like almost like synthetic proteins
Mm-hmm.
in your computer,
Yeah,
essentially meaning that there was like different values of like key parameters
parameters,
like
exactly.
half-lives and amino acid
And then we did
numbers and this.
statistics on those synthesized proteins,
Yeah.
and we could
Hmm.
quantify how many move into the dendrite, how many
Hmm.
mRNAs of those should be moved into the dendrite based on the energy calculation. And then we could
Hmm.
predict particular threshold values as a
Yeah.
function of the dendritic length. We found out
Hmm.
that for longer dendrites, more
Yeah.
mRNAs should be dendritically localized than
Hmm.
for shorter dendrites. That's an experimentally testable hypothesis that we are
Hmm.
now trying to
Okay.
evaluate.
Yeah. Also that you that longer I mean you have like long half-life also makes
Exactly.
the so it is long protein half-life, then you prefer, what do you want to prefer then?
For
Then you can make more in the soma, right? Because it
exactly,
doesn't
like viewed at it from a different perspective. If you have a long-lived protein, it lives
mm,
for multiple days.
sure,
It has enough time to explore all synaptic
exactly.
locations to diffuse
Yeah,
up and down the dendrite. It
yeah, yeah,
doesn't need to be synthesized in the
exactly.
dendrite.
So it's a car, not a pizza.
It's a car, exactly. It's going down everywhere.
Yeah, it doesn't really matter. It doesn't really decay in a matter of days
Yeah.
like a pizza does. Okay, so but also, and then if you have a long mRNA half life, if that lasts long, it then it maybe makes more sense
Sense
to
to move
have.
it, right?
Yeah,
So imagine you have
and
a short-lived, super
yeah,
short-lived
yeah,
mRNA. You
yeah,
invest energy in moving it to the dendrite, and it's gone
yeah, from the soma,
from the
exactly, because
soma.
they all mRNA are have to be produced in the soma. Yeah.
So intuitively, it makes sense that the longer mRNAs
Mm. Yeah.
are, are those that are moved into the dendrite, because they are um, when they arrive, they actually have some time to spend on the synthesis.
Mm.
If
Yeah.
you move short-lived mRNAs, yes, you can do
Yeah.
that mathematically in the model. It works,
Yeah.
but you don't get a lot of it. You don't
Mm.
get the benefit.
Mm. Yeah. So, and I guess you show there's some like in the paper you describe sort of how like some of this has been seen in different, especially if you've seen in experiments.
Yes, we
So
had a lot of fun looking into the
yeah,
data along each
yeah,
parameter dimension. We
yeah,
derived certain things that we thought could be testable experimentally, and
mm,
then we went one by one to
yeah,
look at the data.
yeah. It's a very rich model in that sense.
Yeah,
It
we
has a
spent
lot of
years hunting down the data sets and
yeah,
combining,
yeah,
cross-checking. Some
yeah,
mRNA data sets have one set of genes, and the other protein sets don't match the same species. So that's an interesting
oh,
problem to match,
yeah,
to pair, and to make sure that you are pairing only within the same species. For example, if you connect the red data to mouse data, you can do that, but then referees
mm,
might not be happy. So there are many considerations to take into account.
mm. So exactly, and then so, but if you have then say this one protein like CAMK alpha two or
Hmm.
no CAMK
KMK2.
two alpha,
Hmm.
yeah, CAMK two, then if for some reason the neuron need much more of this, it doesn't really just the number of what you call the copy number doesn't really affect the distribution or the this the well the soma production versus dendrite production that much,
Hmm.
right?
Um, so the model, as we published it, makes
Mm,
predictions for the steady state. The
mm,
dynamics is something interesting on top.
mm.
If you
Okay.
ask the question, how long would a protein take to arrive at my synapse that's now undergoing
Mm,
plasticity? If
mm,
you start to synthesize that protein from scratch, it's better if the mRNAs are already in the dendrites, right?
oh,
The time to
yeah,
arrival is shorter.
yeah, ah,
However, there is one caveat here, and
mm,
that is if this particular protein was synthesizing mRNAs,
mm,
it was localizing mRNAs in in the soma, then there
mm,
would be more in the dendrite to begin with,
okay,
because
yeah,
they have to walk through the dendrite to get to the synapse. There's no other
okay,
way. That's the
mm,
tunnel that gets them to the synapse. So if there are
mm,
many already in the dendrite, you can potentiate also right away. You can source
okay,
from the pool, but
okay,
it's good for the short term. But at some point, the pool may not be big enough, or you
mm,
may need more than the pool affords you. That's and also you need to calibrate the pool again if one synapse takes up more than it should all the time. So there is
mm,
a calculation about how to replenish the pool. That's an interesting theoretical
yeah,
question. But also,
yeah,
if there are many benefits to be explored for the synapse if the protein is synthesized on site, because you can keep synthesizing on top. You already
mm,
invested the cost for the mRNA. So as long as
mm,
that mRNA lives on site, you can synthesize the proteins quickly, relatively quickly, to serve that
mm,
extra need. And all of
yeah,
these strategies need to be evaluated per protein and per time
mm,
constant where you need those proteins. So it's a tight
mm,
balance
mm,
per species involved in the
mm.
plasticity events.
But sort of like the factories ribosomes, right? So they are this. I mean, the ribosomes, the same ribosomes produced all these different proteins, right?
Yes.
So it's really the protein that requires most energy to make. Maybe like because you need a lot of them.
Mhm.
That's maybe what determines where the ribosomes will be.
Yes, it's actually not so easy to calculate the distribution of ribosomes as a function of
Okay.
distance. We are just now
Okay.
starting to do that quantitatively.
Mm,
Many ribosomes are in the soma, but we also know that they are located along the dendrite. But whether it's an
uh,
exponential distribution like it is for the mRNA, we
mm,
don't know. We know that the mitochondria are exponentially distributed. We know that mRNAs are exponentially distributed. It's
mm,
plausible that the ribosomes are also exponentially distributed, but we
mm,
haven't fitted
mm,
the data yet.
mm. Yeah. Cool. So this seems like this have opened up a whole program.
Exactly. We just keep discovering things
Yeah.
that we find interesting. Yeah.
Uh,
And now people
because
also
it's
approach
really
us to have dead particular data sets analyzed in this mathematical way. Yeah.
Yeah.
If you are a young postdoc or a young student listening in, maybe
Yeah,
you should consider the molecular
absolutely. Yeah.
molecular evolution and molecular dynamics as
Mm.
your next
And,
topic.
yeah, and I had a previous student actually here in Oslo, and he moved to Bonn to work. I mean, on scientific programming or something, and he seemed to like it there. So it seems to be a
That's cool.
nice place to live, also.
Yeah, and we also help experimental labs analyze their
Yeah.
own data. We
Yeah.
don't analyze everything for other labs, but
Mm.
we provide self help, so to say.
Mm. Because I think you say in your paper that it's like a handful of very abundant proteins. I'm not
Hmm.
quite sure what the handful mean, like five or whatever,
Hmm.
dominate the metabolic cost.
Yeah,
Really, so it's really
yeah, those that
like,
are
but
super abundant. If you have a
yeah,
high copy number, so if you have many copies to produce, that
mm,
alone will set
mm,
your energy budget off.
yeah.
And
So this
mhm,
this CAMK2 is one of them, maybe.
it's one of them, exactly. It's one of
Yeah,
these
and
most
one of like
abundant
the.
proteins. Yeah.
Mm. So, and then, of course, now you have, if you know if you make assumptions or if you look at the experiments and come up with estimates on how many proteins you need, then you can actually make make an estimate for how much energy the neuron needs
Yeah.
uh, continuously
Yeah.
to
That's
sort of to
an
just keep
interesting
its
calculation. Yeah.
yeah, and then you end up that is actually comparable to the estimates for like for example, the energy you need to fuel the ion pumps that keeps up the ion concentration
Yeah.
across the membrane.
This very famous David Atwell paper that said
Yeah.
the total is about 10 to the power of 8.
Okay.
But our calculations, we can meet 10 to the power of 8 for a single protein species and even
Okay.
more. There are some species
Huh.
in our parameter sampling that would
Mm,
get 10 to the power of 10 ATPs per second to maintain their average
mm,
copy number. That's a lot.
uh, yeah.
And if you think about all of the other 10,000s, you need to sum up their energy budgets, right? So
Uh,
you easily arrive in the same ballpark as the
mhm.
spiking cost. But spiking cost, as the energy for spiking has been used as a motivation to derive theories that
Mm,
minimize the spiking cost. We all know that number of spikes
yeah.
for algorithm need to be kept small, but
Mm,
that same logic hasn't been carried through to the molecular or other
mm.
applications in neurons.
But also, I mean, if say it turns out that the cost of just keeping the the proteins at the right place and at like you have computed, if that is say larger than the much larger than the cost of a spike, then this all this idea about what is called efficient coding and
Coding,
sparse coding, and that's not so important for the neuron
yeah, but those
to
theories have been super useful, right?
yeah, they have,
Even in
yeah,
the
well,
Hopfield model, the minimal energy, that's where the memory is. So if you want to
yeah,
remove that from all theories, I don't know where we get.
but
Um,
that's true, but that's more like in abstract energy, right? The energy function, so it's not physical energy in the same. It's a different kind of energy, isn't it?
it's a
Yeah,
different kind, but is there a mapping between the two energies?
yeah, yeah, that's yeah.
Yeah. Also,
So
there are several theories that say you want to position a spike in a very efficient way to maximize information or to maximize prediction
mm,
quality. But if there are
mm,
enough spikes, position them anywhere.
mm, mm. So so this is sort of cool. I mean, there's also estimates for the like the energy consumption making the synaptic vesicles, and that's also very high. So it seems like the this is the I guess the jury is still out on. We need to find out more about these different energy consuming processes and how they really compete
Hmm.
in the brain. But at least it's sort of it's interesting that
Yeah, we have one new player basically who
yeah,
competes for energy.
for the same energy, yeah. But it least sort of like interesting that you seem like, because using your energy optimization principle, you seem to end up with distributions and solutions which are which are optimal or quite or quite good in terms of reducing energy for just keeping this molecular machinery
Yeah.
going. So that's, at least if it was really really small compared to other things, you might think that why should you bother with optimizing that if
Mm.
the other expenses are much higher? But at least they seem to be in the
The same
same
ballpark.
ballpark.
And
Yeah,
in some
exactly.
instances, it's higher, in some instances, lower, but you certainly cannot neglect this. This is a big
Mm.
expense.
You also have some sort of uh, you get sort of you said that the model doesn't really predict sort of where the ribosomes are, but it has some predictions of where the ATP producers, the mitochondria,
Yeah.
are, right?
Yeah, because if you look into the biochemistry of where energy can be produced in the cell, there are more processes than just the mitochondria.
Mm,
So the question is, if you need an energy item, which of these processes will supply it to you, and how
yeah.
is it balanced?
Mm,
And that's an interesting question. We are now looking into how the ribosomes and the mitochondria, whether they co-localize, and
mm,
whether both have the same spatial distribution. There are many things we don't know yet.
mm,
If
mm,
they were following exactly the same spatial tapering into the dendrite, for example,
mm,
that would be interesting, whether they truly co-localize or whether they are both happen to have the same distribution.
mm, mm. There's one thing which I like about, and of course this is one level below like the typical modeling of electric signaling, and here the parameters are maybe I'm like diffusion constants of mRNA, for example. That's maybe something which is more conserved. I mean, that's actually a number that reflects more. That's really how many. I mean, that you should think that is something that is more comparable across between different neurons.
Mm. And it turns
So
out we calculated the diffusion coefficient that you would expect if
yeah,
the mRNAs were diffusing.
yeah,
There are these Einstein equations where you put in
mm,
the mass, you put in the viscosity of the fluid, and then you get the
yeah,
diffusion coefficient.
yeah.
And those are not terribly off.
Okay,
Of
yeah.
course, some mRNAs are actively transported, right? Then the diffusion coefficient will be
Mm,
different. But if this motion in the dendrite was just
mm,
passive, it's already
yeah,
a reasonable diffusion coefficient.
yeah. So you could even imagine that you use sort of like the distribution of proteins and mRNAs to get estimates for diffusion coefficients. That would be really cool.
Yeah,
I think
yeah.
that would be very
We will,
elegant.
in one of our papers, we collected all of the diffusion coefficients that have been measured in the literature, the generations of students who scored
Mm,
the literature. And we have a
mm.
table containing about 30 species. And then we made a distribution out of that. And
Okay.
that's where we will sample from.
Yeah. Cool. Yeah. This opens up for so many things. So, but you also had this what's called a shank 3.
Mhm.
All these proteins don't have very systematic names, do they?
Yes,
Because
it's
some are short
historically,
names, yeah, long names, and it's just yeah. So shank 3 is a protein, right?
yes, it's a synaptically it's a protein family that is
Mm,
important for both synaptic plasticity and for the anchoring of receptors.
mm, hmm.
And there was a nice
Yep.
data set from the that's published about 20 years ago now, showing how fluorescently labeled Shank3 proteins are moving into the spines. And
Mm,
we really like that data set from Noam Ziv's lab, if I'm not mistaken. Um,
uh,
then, and we wondered whether we can match the time constant of that motion. And it turned out we
mm,
can.
mm,
These proteins arrive at different spines that are located differently from the soma. And we could
mm,
characterize this increase in the fluorescence as a function of the distance. And we were really surprised that our parameters that were not fitted to that particular data set, but just happened to be correct
uh,
biophysically, could also reproduce that temporal evolution.
yeah. So then you use the not the steady state version of the model. You don't put the left hand side to 0.
Exactly. Here we didn't put the left-hand side to 0, but
Mm.
provided a source term and
Yeah.
then looked where these fluorescently labeled proteins are
Mm.
making their way to
Mm.
the synapses. How and how long they take to arrive at the synapses. Yeah.
Yeah. So, how do you think? I mean, now I guess a lot of these data that you compared with comes from hippocampal
Yes, that's
pyramidal
correct.
cells, right? So this, but I guess this maybe more reason to believe that this particular principle is applies to all cells, and that in that respect, I would think that cells are quite similar. You would think,
Hmm.
but so you're waiting for future data sets
Yeah.
and other neurons and
Now, presenting this work at several conferences, several people
yeah,
approached us with exactly these
yeah,
types of data sets in different cell types. And transcriptomics and proteomics has been
mm,
done in different cell types. Particularly, the
yeah,
transcript characterization is really wonderfully done across different cell types. Both single nuclei data is available, single soma
mm,
data, and also whole cell data. In some instances, even patch-seq data of human cells.
mm.
So there is really a wonderful development of theory
Yeah,
that can start.
so what is patch seek data?
Patch-seq is the opportunity to patch a cell and then to
Mm,
label each individual mRNA or close to each individual mRNA in that particular cell that you patch. So it's not just
okay,
average that you calculate, maybe
exactly.
by cutting up tissue, but in that particular cell.
So you got it from like on a single individual neuron basis.
Exactly.
So this, yeah. So do you think that? I mean, there are sort of, I mean, uh, there are like diseases, obviously, like Alzheimer and or
Yeah.
like or ALS. Do you think this kind of, I mean, could it be that, or yeah, that this sort of
How
like
this can be
this,
useful, right?
yeah, and how sort of whether it's sort of like the where the neurons are affected by some like some distribution of molecule disease,
Yeah.
maybe because they lack energy or something,
Yeah.
or don't get,
So that's you're basically following my grants, right? you
yeah,
you see where we are evolving.
yeah,
But these are logical questions, right? This is the most interesting, the
mm,
most impactful direction here. Um,
mm,
what we know is that transcriptomes are different in disease, and they're
mm,
different in aging. They're different across
mm,
cell types. And
mm,
it's really hard to pinpoint what exactly is different because, by the looks
mm,
of it, every transcript is up and down regulated. So,
yeah,
so statistical quantification about which proteins and mRNAs are upregulated how, and what is the
mm.
biophysical reason for them to be upregulated just this way, that's where the interesting questions lie.
It's also be a lot of like quite different during pregnancy, also,
Yeah.
as I've understood it, like all kinds of like the yeah,
Yeah. We
things,
also
yeah,
know that in aging, the machinery to produce mRNAs, not produce mRNAs, produce
mm,
ATPs is impaired. Um,
mm,
and therefore, there might be an energy dearth in the aged tissue. And therefore,
mm,
there might be other downstream effects of that problem
mm,
percolating even to the mRNA localization level. There are many
mm,
questions that we don't yet understand, but we see these phenotypes. The distributions
mm.
look by the eye just already different. You take the histogram
Phenotypes.
and then they
What is
move up and down.
what is phenotypes?
A
Just
phenotype
like,
means um, the there are phenotypes across different levels, right? A phenotype can be on the behavioral level when the
yeah,
animal
it is sort of
behaves
what an animal,
differently,
yeah,
but there are phenotypes also on the molecular level where this number you can define them along different parameters. But for
yeah,
me, it's about the expression level of proteins and mRNAs being different.
yeah,
The
exactly.
histograms are
So,
different.
yeah, exactly. So it's actually some sense like the product is sort
The
of
end
like another
product is
different. That's like, yeah,
yeah.
yeah. So so now
Maybe if I
there's
may add
many,
here,
yeah,
often when we think about a disease, we are hunting for a particular gene that would
mm,
explain this phenotype. But we also
mm,
know that in most cases, it's not a single gene situation. or single
mm,
protein situation. And the moment we are dealing with everything or many things up and down regulated in very strange ways, it's really hard to quantify. And
mm,
that's where I think theory at the population level of transcripts and proteins can really make a dent in our
mm,
understanding.
mm. So, I mean, there's you mentioned this that there are people approaching you with well, experimental data,
Hmm.
but I guess also ideally you would like to sort of to be able to design experiments and like to so
To
that you can
test.
causally check predictions.
Exactly. We
So what
also,
would
once
be
we
yeah
establish rapport with a particular experimental group, it's usually a
yeah
back and forth.
yeah mm. Yeah. So yeah. So cool. So okay. So what would be okay? So maybe so what if you could sort of design an experiment here exactly now?
Hmm.
What would you what
What would
would
we
you
do?
tell your
I would
if you had your
obviously
own
go for the
experiment?
whole brain level, you know.
Yeah,
I
yeah,
would
no, that's true.
look at everything simultaneously, protein, mRNA,
Mm,
all different cell
mm,
types, different species, and see how everything changes, right?
mm. But it's I think this is I mean I used to do like semiconductor like physics, right? And then of course like you always have these people specializing doing experiments, have experimental labs, and and then you had like obviously people doing theory. But there, I at least when I went to good experimental groups, I had a feeling that the PI also, even though he or she didn't do modeling, they he or she sort of knew about like the leading theories.
Hmm.
So they actually sometimes designed the theories or designed experiments
To
in
test.
order to test theories.
Yeah.
And of course this is for because experiments is a big investment. So I mean time and resources and everything. So uh, and I have a feeling that like historically, I think the theoreticians that like the data there's very few experiments that has been made in neuroscience that has been in order
Mm.
to test an experiment, right? Because then
Hmm.
often you need another kind of experiment. Maybe you want sort of like you need. I think often experimentalists naturally they would like because it's difficult to do the experiments, maybe keep like
Mm.
an animal alive for them required number of hours and so on. So often they design the experiments to get the answer to their particular question
Hmm.
as soon with as little experiment as possible,
Hmm.
which is sort of natural, right?
Yeah.
So this thing of designing experiments in order to test to so that you can test models, that's something which
Yeah.
so I mean you are now like a PI, so if you run an experimental lab you could sort of you could actually sort of make these kind of experiments, right?
Hmm.
But you have you considered to branch out to experiments or
Yeah. People keep asking me this, but
yeah
for me, I want to have the freedom to choose different topics.
yeah
Um, and some experiments are um, that, for example, if I was now to follow this energy minimization direction and protein and location
mm,
of mRNA problem, these
mm,
types of experiments would be very diverse across different different methods. I would be interested maybe in patch-seq, or I would
mm,
maybe do SciLac experiments that measure the
uh,
half-lives. And ideally, I want all of it. And that's not something that a single lab can provide. So even if I was to design a single experiment in my own lab, it wouldn't be
yeah
enough. Therefore,
okay
I would still branch out to colleagues and ask,
okay
can you
so
can you check this and this?
exactly
And by now
so
we have we are really living in a really unique time. There are many databases that collect exactly the
mm,
type of data that is
mm,
necessary to test these theories. We have the Genelia databases. We have the Allen Institute. We have the iBrains. We have many eBrains. Sorry, we have
mm,
many large scale efforts to collect data sets that are directly amenable to theory.
mm,
That was their
mm.
original goal. But then
Yeah,
more theories should try to use these
yeah,
data sets.
yeah. And I guess particularly at your at the level of your modeling, there is particularly a lot of data, which is all these new measurement techniques,
Yeah.
and the techniques, or the data is maybe more easier to. There's like there's more. I mean, more reliable in the sense that there's less variation between labs, and
Mm. There
maybe
is
that is
still
more.
variation across labs, I would
Yeah,
say,
yeah,
but there are also ways to normalize for this variation.
yeah,
Detection
yeah.
frequencies are not equal. The
Mm,
gene names that people care about are not equal. The
mm,
overlaps are not. Sometimes they are bigger, sometimes they are smaller. So that's something to care about, um,
okay.
and also to average across labs
But I thought it were sort of like some of these chips they use. That's sort of is not made by
by
you and some
companies.
companies.
Yeah.
Yeah, so that at least they use the same chips, I guess,
Yeah,
more or less.
I'm a theorist. I don't know
Yeah.
exactly
No,
which
no,
chip,
yeah,
but
exactly.
also I know that this is a highly dynamic environment. There are
Yeah,
many companies jumping on these opportunities, and I've learned just
yeah,
recently that there are also companies trying
yeah.
to sell not only the device to record, but some analysis on top.
Well,
And this is direction that's definitely expanding. Maybe
yeah.
somebody wants to do a
This
startup
sounds like
uh, disease
a branch
detection
of,
from transcriptomics.
yeah. This is a branch of neuroscience that seems to be more more reproducible, more likely to get reproducible than say, and like looking at behavior of animals, and you can sort of understand why this is sort of
Yeah.
more difficult to do than
Um,
to go
I
to have
think
lab the same.
it depends on the particular experiments, right? And
Mm,
also on the carefulness of that particular setup. But in general, people do an amazingly good job controlling for many variability, from a
mm.
lot of variability that could occur. Um, and at every level, there are some pros and cons, basically, to
Mm,
every data set that you encounter. And often when you download a data set or you get it from your collaborators, you still need to do post-processing. It's not at face value
mm,
that you can directly analyze. There are still normalization steps that you need to implement yourself oftentimes.
yeah. Hmm. I guess one I guess with this CRISPR technology, you could in principle then like take your proteins and then add say like these sort of like non-coding regions,
Mm,
and then sort of keep almost everything the same except this added non-coding
yeah.
region. And so there's all kinds of
Yeah, there's like a
possibilities for.
sandpit, right, to play.
Yeah,
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 I'm trying also to understand computationally what difference it would make to have a shorter non-coding sequence versus a longer one. Um, is there any patterning? There are also mRNAs that don't have a coding region; they're just regulatory. How can
yeah,
we deal with these types of statistics that some are coding, some are non-coding, and they all interact with one another?
uh, mm.
Yeah, there's some deep theory still to be discovered.
I can imagine, and of course the data is also is like information rich, so you also need to
Exactly. And mostly it has been
understand.
characterized using tools that are more statistical, more
Mm,
descriptive. And I think
mm,
if we want to have a deeper understanding, there needs to be some more principled approach inserted here, like
mm,
the leaky integrated fire, LAPIC type of work, but now for
mm,
the molecular domain.
mm. I mean, if you sort of look at the whole field of neuroscience, I guess I don't know what fraction of scientists would characterize themselves as computational neuroscientists. Maybe like a few percent,
Mm,
maybe. And then of those, how many, what fraction are working on like energy
yeah,
and the energy aspects?
very
That's like
few.
very few.
Very, very few. I think from the molecular perspective, the largest cohort of computational neuroscientists is
uh,
trained to analyze cellular data,
uh,
calcium imaging, patch clamp,
mm,
electrophysiology data, even behavior.
uh,
But the moment we go into more molecular and cellular recordings, ion channel dynamics in cells,
mm,
it's harder to because I think the worry is that there are so many variabilities that you
mm,
could miss. There are just so many
mm.
parameters. There's so many techniques. It's just overwhelming.
Yeah. And when you say computational, I mean there's of course a lot of people with a like a maybe computational neuroscience training that works on like statistical, purely statistical analysis.
Exactly, like data
But the ones like
analysis and
data,
data
but like
process. Yeah,
the kind of what I call mechanistic modeling that you do, right?
mm.
I mean, it's like this is like you're
I think we
starting
need
with.
more of it.
I agree. So that's because those are really really few. I don't know. I mean, yeah, it's so there. I guess you would
I always
recommend or
try to motivate
yeah,
the
I
younger
think
crowd to
that's
keep join us. Not
yeah,
just
and I
my
think
lab. Maybe you want to start your own lab.
yeah, no, I think that's a very and especially now with I think your model is sort of like a little bit of a quite pioneering, and it's a nice breakthrough in sense of making these kind of. I don't have an overview of this field. Maybe other people have done nice work too, but or along these lines, I just don't know it. I just sort of like it was like an epiphany reading your paper about this. There are also models more on the I was like the well, I guess they call it systems neuroscience, but it's really about all these signaling pathways
Yeah.
with this molecule making that. We had a podcast with Polina Shitchkova.
Hmm.
Uh, so it seems to be a Russian thing. You're also Russian or originally
No,
or
I'm Ukrainian. It's
Ukrainian? I'm sorry. These should not be mixed these days, I guess.
exactly not
But
these
anyway,
days.
anyway, so that's okay.
Ukrainian
Anyway, so
by origin, so to say. Yeah.
it's a Slavic thing, maybe. Yeah.
I've
Anyway,
spent my career, my schooling in Germany to a good extent.
okay. I'm sorry. So anyway, so this is, but that's sort of like at an even deeper level in sense. It's sort of is then it's very information rich and it's like all these parameters that you may maybe need like hundreds of differential equations
Yeah,
with the
that's the
parameters you
question.
need to.
I think it still needs to be debated. If we want to capture all of these signaling cascades between different proteins,
Yeah,
do we really need an interaction term per protein
yeah,
pair? Or
yeah,
maybe there is
yeah.
a bit more statistical, maybe correlational analysis that we can
That's
do,
true.
because we
Yeah,
cannot have there are
that's true.
tens of thousands of them. It's hopeless
No, that's
if
true.
you
But
want
it's
to have
also I would say
mechanistic
that
interaction model.
that's true. But also, if you have then like this, I guess Polina's model has like 150 differential question. If you have that sort of like as like a very detailed model and sort of like a ground truth for developing and exploring these more coarse grain models,
Mm,
right? These models are even though they are difficult to understand and even if you don't know all the parameters, they can be very useful for like testing
mm,
these coarse grain theories,
yeah,
right? So it certainly has a
yeah. We need
role
to
there, even
connect
though
them. We need both ends.
exactly we need to connect them exactly.
Yeah,
And often it's easier to connect to a more detailed model than to experiments, right? Because a detailed model can be useful even though it doesn't correspond really to a
yeah.
particular experimental system.
My
Maybe.
philosophy about model building is also to say um, I want to go a little bit one step more than just describe what I find. I want some new effect. I want to be surprised by some measure of surprise.
Yeah, you want to looking for some
I'm
principles that
I'm
tells
always
something.
looking for some principles.
Yeah,
That's
I think
right.
that's
That's
I very I
the
think that's
physics at heart.
exactly that's the physics idea. We hope for principles, and if it's just sort of if you
The spherical
just get
cow.
bogged down in really really complicated numerical models where you can only produce data without really understanding what's going on, it's not so satisfactory.
Yeah.
It at least we hope for something more. So one thing that you told me that sort of you're also quite interested in is sort of like is this innovation aspect on how to take
Hmm.
how to get like innovative sort of like I guess like maybe companies
Hmm.
or stuff out from activities in a scientific lab.
Yeah. Um, what I encountered running my own lab, and I'm sure other people have seen this too, is that you start off your lab with one idea. There's maybe a series of papers pursuing that. And then the question is, do you want to do that for the rest of your life? Or do you want to branch out? And
Uh,
like from my educational upbringing, I was working with correlations of spikes, pairwise
uh,
interactions at the circuit
mm,
level. I fit data to response curves. And at some point, you need to develop a new direction. But
uh,
you are always leaving behind a zone of comfort, right? Where you know everything, where you know the community. So the moment you
mm,
step out is a risk.
Mm.
And you need to build new methods from scratch. You need to get accustomed with a new community from scratch. You need to look into the data sets that are trustworthy, others may not. So it's a really a risk, going into a different direction or adding a new direction. But also we know that technical development is now really accelerating. LMMs and other things are really catching up with us. So there is really a necessity to innovate even within a single lab. We need to take risks. And I was
Mm.
thinking about how to do that in a way that you don't run out of funding or
Mm.
don't impose too much risks on your students. How to
Uh,
do that in a way that's really sustainable and adds a benefit, right?
mm.
Um,
So
and you don't
innovation for
know
you,
which ones of these ideas will be successful when at the start of it, right? So you need to
you
seed
don't
out a few to see which have a potential.
you don't mean actually innovation in terms of making a company.
I
It's
never
you're thinking
started the
more.
company, so I don't know how that really
No,
works.
no, exactly. So you're thinking more about changing your
In a
research
scientific
approach.
lab, in a
Yeah,
scientific
exactly. Yeah, yeah.
lab that
Well,
that needs to think about long-term development, not just grant to grant, but or PhD to
uh,
PhD, but a bit more
okay. I have
long-term.
from back in the days. I have some experience because I switched from like solid state condensed matter physics to
Hmm.
neuroscience,
And how did you
and
do
which is
it?
a big no, no. This was I can tell you because I was a postdoc in San Diego, and then I got this job at like essentially associate professor at the northernmost university of the world in Norway, University of Tromsø. And what is about Norwegian positions, like academic position, that you don't have this tenure track thing.
Hmm.
So the first job I got was like not sort of like an assistant professor. No, it was sort of an associate professor, meaning it was permanent.
Oh, that's good.
So then, exactly. So these are great. So of course, this is like this. Yeah. So that's the system. That's the Norwegian system, and I think also has been like the Scandinavian countries. So then, of course,
You could have
then
stayed,
I
right?
could,
You could have.
yeah. So then I decided to switch because I was more interested. But also then there was this other thing because it's for one thing it was I didn't really get funding for a PhD student, and that's a bit risky, sort of to start off without knowing that you're actually doing something that can get sort of like published or
Mm.
like especially for a PhD student.
And try applying for a
Um,
grant without preliminary data.
exactly. So this is sort of. But then what I had like access to master students, and master students are. I mean, it's like you can have a make a. I mean, a master student can write a very nice thesis without it necessarily getting published,
Mm.
right? I mean, it's
Yeah.
so it was sort of a nice way to actually. So I had like we started doing projects that sort of like with master students.
Mm.
So that was sort of like a training for me to sort of to learn about different. So I had like a period of a few years where I only had master students, also because I didn't get successful. My grants proposals were not successful, so that helped me to sort of to find a subject where you sort of felt that now I can supervise a PhD student
Mm.
and without sort of risking the career of the PhD
Yeah, I
student,
did it
right? So
exactly the same way with master students. There
yeah.
was one
Mm,
influential neuron paper that we published 6 years ago now,
mm.
2019, I believe, where the master student is the first author for exactly that same reason. Yeah.
Yeah, we have actually that. Yeah, that's quite common in our group that it sort of starts with a master student, but then of course it has often needs to be some more work done. So then we have to yeah.
Yeah, exactly.
So I also they talk about different experiences because then when you start with a master project, then that's well if you find a master project,
Mhm.
often a typical thing you do is that you find a model that you think look interesting or is on the subject you're interested in, and then you want sort of like the master student to build on that maybe and explore some new avenues or something. And then we started with this one model. I remember there was sort of like this very complicated like the models of dendrites in an interneuron. And then it was just impossible to reproduce. It was sort of, I mean, the information was there, but it sort of didn't really make sense. There was some kind of bug in the description. So then I remember I wrote to the person who made this model,
Mm-hmm.
and he got upset and said, "Well, I can't find the binder, and anyway, you should make your own model. You shouldn't use my model." So this, which was sort of a very different culture, right? Because in physics, you're very happy if people decide to choose your model and try to build on that, right?
Mm.
Because there are alternatives and models out there,
Yeah.
right? Which they could have chosen. So this I just realized that this is the culture. Culture was quite different. I think it has. This was back in the late 90s, so I think things have sort of the culture has sort of has changed.
Mm.
So I don't know if you saw this story that John Hopfield wrote about. He used to do some work on some like biochemical modeling, and he wrote this story in one of his
Yeah.
papers about when he was attending this conference, and one of these seniors and was sort of like modeling some data he had looked he had found, and a person took him aside, and a senior guy said that this it's not really good style to work on other people's data, right?
Mm.
So it's like completely different, which is sort of like the very different from the physics culture, where it's sort of like where the certainly the experimentalists sort of produce data that sort of like modelers try to, or theoreticians try to understand, and they all share
Mm.
glory and Nobel prizes and so on.
Yeah.
So I just realized that there are sort of like cultural
Mm.
differences between fields, but I think this like the general attitude towards modeling has improved, or at least from my
Yeah.
perspective,
There are just
has
more of
changed
us,
quite.
right? The
Yeah.
N is
I
increasing,
also,
and
yeah,
the
and I
variability, and
also
it's a
think
community now.
yeah, and I also think that like good experimental also realize that sort of modeling can actually add a lot of value to their data and to their sort of, so they want to,
Yeah.
yeah. So things have
Yeah, and the beauty of the theories, I don't need to to motivate that for you, but
uh,
you can connect different levels, right? You can take
mm,
mRNAs with proteins. You can go to synaptic dynamics. You can add a circuit level to it. You can
mm,
take different data sets from different labs and say, hey, here's a connection. Have you thought about this?
yeah,
And that's not something that usually comes up in your mind when you are working on one particular
yeah,
technique, on
mm, mm,
one particular molecule even. Yeah.
mm. But now I told you sort of my story how I switched. So what
What's my story?
so
Yeah.
what is, yeah, what's your story here?
um, yeah, so when we discovered that there are these really nice data sets characterizing mRNAs and proteins, the question was, how do I add that dimension to my lab, right? I
Mm,
cannot just say, okay, we're going to stop this and we are going to add this particular direction and everybody should pivot here. No, that's not how
mm,
it went. There were a lot of exploratory projects, also master students project, and some worked out, some didn't, or some evolved to a point where things were interested or getting interesting. And there was a lot of trial and error in between. And there were some interesting breakthroughs along the way that set the direction for the next projects. And
mm,
I didn't submit for the first grant before we published one of these papers. Um, when we
mm,
submitted the first paper, when we were through the review, that's where I felt comfortable submitting a major grant on these topics. But
mm.
before, I thought, okay, nobody will believe me if I do.
No, no, you don't have the
Yeah.
credibility and standing.
Yeah. And in my own lab, I try to encourage people to choose side projects because for
Mm,
that risk minimization, obviously, but also to
yeah.
explore different directions. If somebody is working primarily on the molecular dynamics that we discussed,
Mm,
maybe it's cool to look at connectivity, circuit level addition to
mm,
what you do, or maybe start something a little bit off topic to see if it's interesting. Just be a little
mm,
bit more curious than just
yeah.
narrow it in into particular model,
Mm,
because the
yeah.
curiosity will eventually drive the number of your publications up, but also connect you to different communities, and you will just enjoy your work more. I believe.
Yeah. Yeah. And of course, an interesting thing with modeling is that, I mean, you ended up with a very standard physics type equation, partial differential equation.
Yeah.
So suddenly you have all these tools that we can, well, you know yourself already, or if not, you can sort of dig it up in the literature.
Mm.
So this is also
Yeah.
a particular, I guess, for modelers. These are
The question is always which equation to apply to this problem. And there are students from the math or physics background that come to my lab and they say, "Oh, I want to work on that equation that you published 5 years ago." And I say, "I'm
okay.
not sure I can guarantee that." Because
Uh,
the equations might change, the problem might stay the same,
mm,
but the way we
yeah.
address it, maybe it's more permutation matrices this time, maybe it's more um, I don't know, probabilistic rather than average type of equation.
Mm,
I really
yeah.
try to pick the methods based on a problem at hand. So there is no
Yeah. Mm,
guarantee that that we will work with the linear equations going forward.
yeah.
Maybe there is something
But it's
added.
true. I was sort of collaborated with like I mean, lots of different people, of course there are, and also several mathematicians, and they are quite different. I mean, in the sense, some are like, I would say, like more like maybe like physicists in their approach, and actually focus on the problem, and some just love the equation.
Yeah.
So they don't want to, they don't really want to know too much about why this equation is important. They, so we sometimes call it collaboration through the mailbox. You send in the equations and you get out the answer, and that's probably not the most efficient way. They at least the way I think about it.
Also, in
So
some equational settings, you discover that that one term is small compared to others, so it's going to be dropped.
exactly,
If that's your
exactly.
favorite
That's
term, I'm not sure you enjoy it.
that's true. That you can. No, this is sort of like, anyway. So there are different, certainly different. That's something you learn from switching fields. Is that like there's so many ways to do natural science, also not just like natural science versus social science, which is sort of clearly different,
Yeah,
but also within natural sciences, it's and then
yeah,
well, including mathematics here, and it's sort of like different cultures, different sort of ideas about what's interesting, and so on. So that's
yeah. And
so like I said,
when I was doing my
yeah,
postdoc, my mentor was Larry Abbott, and he's very
mm,
famous for switching fields. You have seen him.
yeah,
I've watched his papers evolve, and I'm
mm,
always surprised what the next paper will be about.
yeah. That's true. Yeah, I guess he also came from particle
He
physics
came
back
from
in the
particle
days.
physics.
Yeah,
Yeah,
yeah. Oh, that's true. So I sometimes say that, like, when I came from like this condensed matter physics and into neuroscience, was a little bit like the first time I left Norway. Like going from Norway to the United Nations. It's like from a monoculture to a very heterogeneous culture, which makes it more interesting, but also sometimes challenging, right?
yeah.
And it's
Also, if you
much more
fail,
fun.
nobody will notice, right? It's less embarrassing.
Because there's so many people. Yeah, so many ways to. Anyway, so this has been fun, Tatiana.
Thank
So do
you.
you have any things that you would like
No,
to
I enjoy
add
talking to
before?
you. Thank you for the
Yeah,
invitation.
yeah, sure. Yeah. Okay, well, thanks a lot for coming to the podcast, and I'm quite sure we'll meet around somewhere.
Exactly. Maybe the Bernstein
Okay,
conference would be the next one.
yeah, exactly. And that's my that's a very nice conference. Every like late this year, it's going to be in, is it Frankfurt? No, no, where it's
I
going
think
to
it's
be?
Frankfurt. Yes.
Yeah, now it's sort of like a nice mixture of experiments and theory, and also for like just this is from like flying from Norway to Germany is so easy. It's like going domestic. It's like no pass or anything. It's like very
Exactly.
practical to be within the, yeah, more or less the more borderless part of Europe.
Exactly. We,
Mm.
we should have
Yeah,
more of those, right? The
I agree. Yeah,
borderless interactions.
yeah. Okay. Well, thanks a lot.
Thank you, and see you around.
Yeah. See you around.
Bye bye.